Hybrid sex, Haldane's rule. Haldane's rule states that when one of the two sexes is absent in interspecific hybrids between two specific species, then the sex that is not produced is rare or as sterile as the heterozygous or heterogametic sex. 40. In mammals, at least, there is growing evidence to suggest that this is due to high rates of mutation of the genes determining masculinity in the Y chromosome. 40, 41, 42. It has been suggested that Haldane's rule simply reflects the fact that the male sex is more sensitive than the female when the sex-determining genes are included in a hybrid genome. But there are also organisms in which the heterozygous sex is the female. Birds and butterflies, and the law is followed in these organisms. Therefore, it is not a problem related to sexual development, nor with the sex chromosomes. Haldane proposed that the stability of hybrid individual development requires the full gene complement of each parent species, so that the hybrid of the heterozygous sex is unbalanced, i.e. missing at least one chromosome from each of the parental species. For example, the hybrid male obtained by crossing D. melanogaster females with D. simulans males, which is non-viable, lacks the X chromosome of D. simulans. 12. Genetics. Precopulatory mechanisms in animals. The genetics of ethological isolation barriers will be discussed first. Precopulatory isolation occurs when the genes necessary for the sexual reproduction of one species differ from the equivalent genes of another species, such that if a male of species A and a female of species B are placed together they are unable to copulate. Study of the genetics involved in this reproductive barrier tries to identify the genes that govern distinct sexual behaviors in the two species. The males of Drosophila melanogaster and those of D. simulans conduct an elaborate courtship with their respective females, which are different for each species, but the differences between the species are more quantitative than qualitative. In fact, the simulans males are able to hybridize with the melanogaster females. Although there are lines of the latter species that can easily cross, there are others that are hardly able to. Using this difference, it is possible to assess a minimum number of genes involved in precopulatory isolation between the melanogaster and simulan species and their chromosomal location. 12. In experiments, flies of the D. melanogaster line, which hybridizes rarely with simulans, were crossed with another line that it does not hybridize with, or rarely. The females of the segregated populations obtained by this cross were placed next to simulans males and the percentage of hybridization was recorded, which is a measure of the degree of reproductive isolation. It was concluded from this experiment that 3 of the 8 chromosomes of the haploid complement of D. melanogaster carry at least one gene that affects isolation, such that substituting one chromosome from a line of low isolation with another of high isolation reduces the hybridization frequency. In addition, interactions between chromosomes are detected so that certain combinations of the chromosomes have a multiplying effect. 12. Cross-incompatibility or incongruence in plants is also determined by major genes that are not associated at the self-incompatibility S locus. 43, 44, 45. Post-copulation or fertilization mechanisms in animals. Reproductive isolation between species appears, in certain cases, a long time after fertilization and the formation of the zygote, as happens, for example, in the twin species Drosophila pavani and D. gaucha. The hybrids between both species are not sterile, in the sense that they produce viable gametes, ovules and spermatozoa. However, they cannot produce offspring as the sperm of the hybrid male do not survive in the semen receptors of the females, be they hybrids or from the parent lines. In the same way, the sperm of the males of the two parent species do not survive in the reproductive tract of the hybrid female. 12. This type of post-copulatory isolation appears as the most efficient system for maintaining reproductive isolation in many species. 46. The development of a zygote into an adult is a complex and delicate process of interactions between genes in the environment that must be carried out precisely, and if there is any alteration in the usual process, caused by the absence of a necessary gene or the presence of a different one, it can arrest the normal development, causing the non-viability of the hybrid or its sterility. It should be borne in mind that half of the chromosomes and genes of a hybrid are from one species and the other half come from the other. If the two species are genetically different, there is little possibility that the genes from both will act harmoniously in the hybrid. From this perspective, only a few genes would be required in order to bring about post-copulatory isolation, as opposed to the situation described previously for precopulatory isolation. 1247. In many species where precopulatory reproductive isolation does not exist, hybrids are produced but they are of only one sex. This is the case for the hybridization between females of Drosophila simulans and Drosophila melanogaster males. The hybridized females die early in their development so that only males are seen among the offspring. However, populations of D. simulans have been recorded with genes that permit the development of adult hybrid females, that is, the viability of the females is rescued. It is assumed that the normal activity of these speciation genes is to inhibit the expression of the genes that allow the growth of the hybrid. There will also be regulator genes. 12. A number of these genes have been found in the melanogaster species group. The first to be discovered was LHR, lethal hybrid rescue, located in chromosome 2 of D. simulans. This dominant allele allows the development of hybrid females from the cross between simulans females and melanogaster males. 48. A different gene, also located on chromosome 2 of D. simulans is SHFR, that also allows the development of female hybrids, its activity being dependent on the temperature at which development occurs. 49. Other similar genes have been located in distinct populations of species of this group. In short, only a few genes are needed for an effective post-copulatory isolation barrier mediated through the non-viability of the hybrids. As important as identifying an isolation gene is knowing its function. The HMR gene, linked to the X chromosome and implicated in the viability of male hybrids between D. melanogaster and D. simulans, is a gene from the proto-oncogene family MyB that codes for a transcriptional regulator. Two variants of this gene function perfectly well in each separate species, but in the hybrid they do not function correctly, possibly due to the different genetic background of each species. Examination of the allele sequence of the two species shows that change of direction substitutions are more abundant than synonymous substitutions, suggesting that this gene has been subject to intense natural selection. 50. The Dobzhansky-Miller model proposes that reproductive incompatibilities between species are caused by the interaction of the genes of the respective species. It has been demonstrated recently that LHR has functionally diverged in D. simulans and will interact with HMR which, in turn, has functionally diverged in D. melanogaster to cause the lethality of the male hybrids. LHR is located in a heterochromatic region of the genome and its sequence has diverged between these two species in a manner consistent with the mechanisms of positive selection. 51. An important unanswered question is whether the genes detected correspond to old genes that initiated the speciation favoring hybrid non-viability, or modern genes that have appeared post-speciation by mutation, that are not shared by the different populations and that suppress the effect of the primitive non-viability genes. The ODISH abbreviation of Odysseus gene causes partial sterility in the hybrid between Drosophila simulans and a related species, D. mauritiana, which is only encountered on Mauritius, and is of recent origin. This gene shows monophyly in both species and also has been subject to natural selection. It is thought that it is a gene that intervenes in the initial stages of speciation, while other genes that differentiate the two species show polyphily. ODISH originated by duplication in the genome of Drosophila and has evolved at very high rates in D. mauritania, while its paralog, UNC4, is nearly identical between the species of the group Melanogaster. 52, 53, 54, 55. Seemingly, all these cases illustrate the manner in which speciation mechanisms originated in nature. Therefore they are collectively known as speciation genes, or possibly, gene sequences with a normal function within the populations of a species that diverge rapidly in response to positive selection, thereby forming reproductive isolation barriers with other species. In general, all these genes have functions in the transcriptional regulation of other genes. 56. The NUP96 gene is another example of the evolution of the genes implicated in post-copulatory isolation. It regulates the production of one of the approximately 30 proteins required to form a nuclear pore. In each of the simulans groups of Drosophila, the protein from this gene interacts with the protein from another, as yet undiscovered, gene on the X chromosome in order to form a functioning pore. However, in a hybrid the pore that is formed is defective and causes sterility. The differences in the sequences of NUP96 have been subject to adaptive selection, similar to the other examples of speciation genes described above. 57, 58. Post-copulatory isolation can also arise between chromosomally differentiated populations due to chromosomal translocations and inversions. 59. If, for example, a reciprocal translocation is fixed in a population, the hybrid produced between this population and one that does not carry the translocation will not have a complete meiosis. This will result in the production of unequal gametes containing unequal numbers of chromosomes with a reduced fertility. In certain cases, complete translocations exist that involve more than 2 chromosomes, so that the meiosis of the hybrids is irregular and the fertility is 0 or nearly 0. 60. Inversions can also give rise to abnormal gametes in heterozygous individuals, but this effect has little importance compared to translocations. 59. An example of chromosomal changes causing sterility in hybrids comes from the study of Drosophila nacida and D. albomicans, which are twin species from the Indo-Pacific region. There is no sexual isolation between them and the F1 hybrid is fertile. However, the F2 hybrids are relatively infertile and leave few descendants which have a skewed ratio of the sexes. The reason is that the X chromosome of albomicans is translocated and linked to an autosome which causes abnormal meiosis in hybrids. Robertsonian translocations are variations in the numbers of chromosomes that arise from either the fusion of 2 acrocentric chromosomes into a single chromosome with 2 arms, causing a reduction in the haploid number, or conversely, or the fission of one chromosome into 2 acrocentric chromosomes, in this case increasing the haploid number. The hybrids of 2 populations with differing numbers of chromosomes can experience a certain loss of fertility, and therefore a poor adaptation, because of irregular meiosis. In plants, a large variety of mechanisms have been demonstrated to reinforce reproductive isolation between closely related plant species that either historically lived or currently live in symmetry. This phenomenon is driven by strong selection against hybrids, typically resulting from instances in which hybrids suffer reduced fitness. Such negative fitness consequences have been proposed to be the result of negative epistasis in hybrid genomes and can also result from the effects of hybrid sterility. 61. In such cases, selection gives rise to population-specific isolating mechanisms to prevent either fertilization by interspecific gametes or the development of hybrid embryos. Because many sexually reproducing species of plants are exposed to a variety of interspecific gametes, natural selection has given rise to a variety of mechanisms to prevent the production of hybrids. 62. These mechanisms can act at different stages in the developmental process and are typically divided into 2 categories, pre-fertilization and post-fertilization, indicating at which point the barrier acts to prevent either zygote formation or development. In the case of angiosperms and other pollinated species, pre-fertilization mechanisms can be further subdivided into 2 more categories, pre-pollination and post-pollination, the difference between the 2 being whether or not a pollen tube is formed. Typically when pollen encounters a receptive stigma, a series of changes occur which ultimately lead to the growth of a pollen tube down the style, allowing for the formation of the zygote. Empirical investigation has demonstrated that these barriers act at many different developmental stages and species can have none, one, or many barriers to hybridization with interspecifics. Examples of pre-fertilization mechanisms. A well-documented example of a pre-fertilization isolating mechanism comes from study of Louisiana iris species. These iris species were fertilized with interspecific and conspecific pollen loads, and it was demonstrated by measure of hybrid progeny success that differences in pollen tube growth between interspecific and conspecific pollen led to a lower fertilization rate by interspecific pollen. 63. This demonstrates how a specific point in the reproductive process is manipulated by a particular isolating mechanism to prevent hybrids. Another well-documented example of a pre-fertilization isolating mechanism in plants comes from study of the 2 wind-pollinated birch species. Study of these species led to the discovery that mixed conspecific and interspecific pollen loads still result in 98% conspecific fertilization rates, highlighting the effectiveness of such barriers. 64. In this example, pollen tube incompatibility and slower generative mitosis have been implicated in the post-pollination isolation mechanism. Examples of post-fertilization mechanisms. Crosses between diploid and tetraploid species of Paspalum provide evidence of a post-fertilization mechanism preventing hybrid formation when pollen from tetraploid species was used to fertilize a female of a diploid species. 65. There were signs of fertilization and even endosperm formation, but subsequently this endosperm collapsed. This demonstrates evidence of an early post-fertilization isolating mechanism, in which a hybrid early embryo is detected and selectively aborted. 66. This process can also occur later during development, in which developed hybrid seeds are selectively aborted. 67. Effects of hybrid necrosis. Plant hybrids often suffer from an autoimmune syndrome known as hybrid necrosis. In the hybrids, specific gene products contributed by one of the parents may be inappropriately recognized as foreign and pathogenic, and thus trigger pervasive cell death throughout the plant. 68. In at least one case, a pathogen receptor, encoded by the most variable gene family in plants, was identified as being responsible for hybrid necrosis. 69. Chromosomal rearrangements in yeast. In brewer's yeast Saccharomyces cerevisiae, chromosomal rearrangements are a major mechanism to reproductively isolate different strains. Hu et al. 70 showed that reproductive isolation acts postzygotically and could be attributed to chromosomal rearrangements. These authors crossed 60 natural isolates sampled from diverse niches with the reference strain S288C and identified 16 cases of reproductive isolation with reduced offspring viabilities, and identified reciprocal chromosomal translocations in a large fraction of isolates. 70. Incompatibility caused by microorganisms. In addition to the genetic causes of reproductive isolation between species, there's another factor that can cause postzygotic isolation, the presence of microorganisms in the cytoplasm of certain species. The presence of these organisms in a species and their absence in another causes the non-viability of the corresponding hybrid. For example, in the semi-species of the group D Polystorm, the hybrid females are fertile but the males are sterile. This is due to the presence of a Wolbachia 71 in the cytoplasm, which alters spermatogenesis leading to sterility. It is interesting that incompatibility or isolation can also arise at an interspecific level. Populations of these simulants have been studied that show hybrid sterility according to the direction of the cross. The factor determining sterility has been found to be the presence or absence of a microorganism Wolbachia and the population's tolerance or susceptibility to these organisms. This interpopulation incompatibility can be eliminated in the laboratory through the administration of a specific antibiotic to kill the microorganism. Similar situations are known in a number of insects, as around 15% of species show infections caused by this symbiont. It has been suggested that, in some cases, the speciation process has taken place because of the incompatibility caused by this bacteria. Two wasp species Nasonia gralti and N. longicornis carry two different strains of Wolbachia. Crosses between an infected population and one free from infection produces a nearly total reproductive isolation between the semi-species. However, if both species are free from the bacteria or both are. Selection for reproductive isolation between two Drosophila species. In 1950, K.F. Koopman reported results from experiments designed to examine the hypothesis that selection can increase reproductive isolation between populations. He used D. pseudobscura and D. persimilis in these experiments. 76. In 1950, K.F. Koopman reported results from experiments designed to examine the hypothesis that selection can increase reproductive isolation between populations. In 1950, K.F. Koopman reported results from experiments designed to examine the hypothesis that selection can increase reproductive isolation between populations. He used D. pseudobscura and D. persimilis in these experiments. When the flies of these species are kept at 16 degrees Celsius, approximately a third of the matings are interspecific. In the experiment, equal numbers of males and females of both species were placed in containers suitable for their survival and reproduction. The progeny of each generation were examined in order to determine if there were any interspecific hybrids. These hybrids were then eliminated. An equal number of males and females of the resulting progeny were then chosen to act as progenitors of the next generation. As the hybrids were destroyed in each generation, the flies that solely mated with members of their own species produced more surviving descendants than the flies that mated solely with individuals of the other species. In the adjacent table, it can be seen that for each generation the number of hybrids continuously decreased up to the 10th generation when hardly any interspecific hybrids were produced. 77. It is evident that selection against the hybrids was very effective in increasing reproductive isolation between these species. From the third generation, the proportions of the hybrids were less than 5%. This confirmed that selection acts to reinforce the reproductive isolation of two genetically divergent populations if the hybrids formed by these species are less well adapted than their parents. These discoveries allowed certain assumptions to be made regarding the origin of reproductive isolation mechanisms in nature. Namely, if selection reinforces the degree of reproductive isolation that exists between two species due to the poor adaptive value of the hybrids, it is expected that the populations of two species located in the same area will show a greater reproductive isolation than populations that are geographically separated. See reinforcement. This mechanism for reinforcing hybridization barriers in sympatric populations is also known as the Wallace effect, as it was first proposed by Alfred Russel Wallace at the end of the 19th century, and it has been experimentally demonstrated in both plants and animals. 78, 79, 80, 81, 82, 83. Selection. The sexual isolation between Drosophila miranda and D. pseudobscura, for example, is more or less pronounced according to the geographic origin of the flies being studied. Flies from regions where the distribution of the species is superimposed show a greater sexual isolation than exists between populations originating in distant regions. Reproductive isolation can be caused by allopatric speciation. A population of Drosophila was divided into subpopulations selected to adapt to different food types. After some generations, the two subpopulations were mixed again. Subsequent matings occurred between individuals belonging to the same adapted group. 84. On the other hand, interspecific hybridization barriers can also arise as a result of the adaptive divergence that accompanies allopatric speciation. This mechanism has been experimentally proved by an experiment carried out by Diane Dodd on D. pseudobscura. A single population of flies was divided into two, with one of the populations fed with starch-based food and the other with maltose-based food. This meant that each subpopulation was adapted to each food type over a number of generations. After the populations had diverged over many generations, the groups were again mixed. It was observed that the flies would mate only with others from their adapted population. This indicates that the mechanisms of reproductive isolation can arise even though the interspecific hybrids are not selected against. Group selection. Group selection is a proposed mechanism of evolution in which natural selection acts at the level of the group, instead of at the level of the individual or gene. Early explanations of social behavior, such as the lekking of blackcock, spoke of the good of the species. 1. Blackcocks of the Lek watercolor and body color by Archibald Thorburn, 1901. Early authors such as V.C. Wynne Edwards and Conrad Lorenz argued that the behavior of animals could affect their survival and reproduction as groups, speaking for instance of actions for the good of the species. In the 1930s, R.A. Fisher and J.B.S. Haldane proposed the concept of kin selection, a form of altruism from a gene-centered view of evolution, arguing that animals should sacrifice for their relatives, and thereby implying that they should not sacrifice for non-relatives. From the mid-1960s, evolutionary biologists such as John Maynard Smith, W.D. Hamilton, George C. Williams, and Richard Dawkins argued that natural selection acted primarily at the level of the gene. They argued on the basis of mathematical models that individuals would not altruistically sacrifice fitness for the sake of a group unless it would ultimately increase the likelihood of an individual passing on their genes. A consensus emerged that group selection did not occur, including in special situations such as the haplodiploid social insects like honeybees, in the Hymenoptera, where kin selection explains the behavior of non-reproductives equally well, since the only way for them to reproduce their genes is via kin. 2. In 1994, David Sloan Wilson and Elliot Sober argued for multi-level selection, including group selection, on the grounds that groups, like individuals, could compete. In 2010, 3 authors including E.O. Wilson, known for his work on social insects especially ants, again revisited the arguments for group selection. 3. They argued that group selection can occur when competition between two or more groups, some containing altruistic individuals who act cooperatively together, is more important for survival than competition between individuals within each group. 3. Provoking a strong rebuttal from a large group of ethologists. 2. Early developments. Charles Darwin developed the theory of evolution in his book, Origin of Species. Darwin also made the first suggestion of group selection in The Descent of Man that the evolution of groups could affect the survival of individuals. He wrote, if one man in a tribe invented a new snare or weapon, the tribe would increase in number, spread, and supplant other tribes. In a tribe thus rendered more numerous, there would always be a rather better chance of the birth of other superior and inventive members. 4. 5. Once Darwinism had been accepted in the modern synthesis of the mid-20th century, animal behavior was glibly explained with unsubstantiated hypotheses about survival value, which was largely taken for granted. The naturalist Conrad Lorenz had argued loosely in books like On Aggression, 1966, that animal behavior patterns were, for the good of the species, 1-6 without actually studying survival value in the field. 6. Richard Dawkins noted that Lorenz was a good of the species man. 7. So accustomed to group selection thinking that he did not realize his views contravened orthodox Darwinian theory. 7. The ethologist Nico Tinbergen praised Lorenz for his interest in the survival value of behavior, and naturalists enjoyed Lorenz's writings for the same reason. 6. In 1962, group selection was used as a popular explanation for adaptation by the zoologist V.C. Wynne Edwards. 8. 9. In 1976, Richard Dawkins wrote a well-known book on the importance of evolution at the level of the gene or the individual, the selfish gene. 10. Social behavior in honeybees is explained by kin selection. Their haploidiploid inheritance system makes workers very closely related to their queen center. From the mid-1960s, evolutionary biologists argued that natural selection acted primarily at the level of the individual. In 1964, John Maynard Smith, 11th C.M. Parents, 1964-12, and George C. Williams in his 1966 book Adaptation and Natural Selection cast serious doubt on group selection as a major mechanism of evolution. Williams's 1971 book Group Selection assembled writings from many authors on the same theme. 13. 14. It was at that time generally agreed that this was the case even for eusocial insects such as honeybees, which encourages kin selection, since workers are closely related. 2. Kin selection and inclusive fitness theory. Experiments from the late 1970s suggested that selection involving groups was possible. 15. Early group selection models assumed that genes acted independently, for example a gene that coded for cooperation or altruism. Genetically based reproduction of individuals implies that, in group formation, the altruistic genes would need a way to act for the benefit of members in the group to enhance the fitness of many individuals with the same gene. 16. But it is expected from this model that individuals of the same species would compete against each other for the same resources. This would put cooperating individuals at a disadvantage, making genes for cooperation likely to be eliminated. Group selection on the level of a species is flawed because it is difficult to see how selective pressures would be applied to competing, non-cooperating individuals. 10. Kin selection between related individuals is accepted as an explanation of altruistic behavior. R. A. Fisher in 1930, 17, and J.B.S. Haldane in 1932, 18, set out the mathematics of kin selection, with Haldane famously joking that he would willingly die for two brothers or 8 cousins. 19. In this model, genetically related individuals cooperate because survival advantages to one individual also benefit kin who share some fraction of the same genes, giving a mechanism for favoring genetic selection. 20. Inclusive fitness theory, first proposed by W. D. Hamilton in the early 1960s, gives a selection criterion for evolution of social traits when social behavior is costly to an individual organism's survival and reproduction. The criterion is that the reproductive benefit to relatives who carry the social trait, multiplied by the relatedness, the probability that they share the altruistic trait, exceeds the cost to the individual. Inclusive fitness theory is a general treatment of the statistical probabilities of social traits accruing to any other organisms likely to propagate a copy of the same social trait. Kin selection theory treats the narrower but simpler case of the benefits to close genetic relatives, or what biologists call kin, who may also carry and propagate the trait. A significant group of biologists support inclusive fitness as the explanation for social behavior in a wide range of species, as supported by experimental data. An article was published in Nature with over 100 co-authors. 2. One of the questions about kin selection is the requirement that individuals must know if other individuals are related to them, or kin recognition. Any altruistic act has to preserve similar genes. One argument given by Hamilton is that many individuals operate in viscous conditions, so that they live in physical proximity to relatives. Under these conditions, they can act altruistically to any other individual, and it is likely that the other individual will be related. This population structure builds a continuum between individual selection, kin group selection and group selection without a clear boundary for each level. However, early theoretical models by D.S. Wilson et al., 21, and Taylor, 22, showed that pure population viscosity cannot lead to cooperation and altruism. This is because any benefit generated by kin cooperation is exactly cancelled out by kin competition. Additional offspring from cooperation are eliminated by local competition. Middeldorf and D.S. Wilson later showed that if the population is allowed to fluctuate, then local populations can temporarily store the benefit of local cooperation and promote the evolution of cooperation and altruism. 23. By assuming individual differences and adaptations, Young further showed that the benefit of local altruism can be stored in the form of offspring quality and thus promote the evolution of altruism even if the population does not fluctuate. This is because local competition among more individuals resulting from local altruism increases the average local fitness of the individuals that survive. 24. Another explanation for the recognition of genes for altruism is that a single trait, group reciprocal kindness, is capable of explaining the vast majority of altruism that is generally accepted as good by modern societies. The phenotype of altruism relies on recognition of the altruistic behavior by itself. The trait of kindness will be recognized by sufficiently intelligent and undeceived organisms and other individuals with the same trait. Moreover, the existence of such a trait predicts a tendency for kindness to unrelated organisms that are apparently kind, even if the organisms are of another species. The gene need not be exactly the same, so long as the effect or phenotype is One of the questions about kin selection is the requirement that individuals must know if other individuals are related to them, or kin recognition. Any altruistic act has to preserve similar genes. One argument given by Hamilton is that many individuals operate in viscous conditions, so that they live in physical proximity to relatives. Under these conditions, they can act altruistically to any other individual, and it is likely that the other individual will be related. This population structure builds a continuum between individual selection, kin group selection and group selection without a clear boundary for each level. However, early theoretical models by D.S. Wilson et al., 21, and Taylor, 22, showed that pure population viscosity cannot lead to cooperation and altruism. This is because any benefit generated by kin cooperation is exactly cancelled out by kin competition. Additional offspring from cooperation are eliminated by local competition. Middeldorf and D.S. Wilson later showed that if the population is allowed to fluctuate, then local populations can temporarily store the benefit of local cooperation and promote the evolution of cooperation and altruism. 23. By assuming individual differences and adaptations, Young further showed that the benefit of local altruism can be stored in the form of offspring quality and thus promote the evolution of altruism even if the population does not fluctuate. This is because local competition among more individuals resulting from local altruism increases the average local fitness of the individuals that survive. 24. Another explanation for the recognition of genes for altruism is that a single trait, group reciprocal kindness, is capable of explaining the vast majority of altruism that is generally accepted as good by modern societies. The phenotype of altruism relies on recognition of the altruistic behavior by itself. The trait of kindness will be recognized by sufficiently intelligent and undeceived organisms and other individuals with the same trait. Moreover, the existence of such a trait predicts a tendency for kindness to unrelated organisms that are apparently kind, even if the organisms are of another species. The gene need not be exactly the same, so long as the effect or phenotype is similar. Multiple versions of the gene, or even mean, would have virtually the same effect. This explanation was given by Richard Dawkins as an analogy of a man with a green beard. Green-bearded men are imagined as tending to cooperate with each other simply by seeing a green beard, where the green beard trait is incidentally linked to the reciprocal kindness trait. 10. Multi-level selection theory. Kin selection or inclusive fitness is accepted as an explanation for cooperative behavior in many One of the questions about kin selection is the requirement that individuals must know if other individuals are related to them, or kin recognition. Any altruistic act has to preserve similar genes. One argument given by Hamilton is that many individuals operate in viscous conditions, so that they live in physical proximity to relatives. Under these conditions, they can act altruistically to any other individual, and it is likely that the other individual will be related. This population structure builds a continuum between individual selection, kin group selection and group selection without a clear boundary for each level. However, early theoretical models by D.S. Wilson et al. 21 and Taylor 22 showed that pure population viscosity cannot lead to cooperation and altruism. This is because any benefit generated by kin cooperation is exactly cancelled out by kin competition. Additional offspring from cooperation are eliminated by local competition. Middeldorf and D.S. Wilson later showed that if a population is allowed to fluctuate, then local populations can temporarily store the benefit of local cooperation and promote the evolution of cooperation and altruism. 23. By assuming individual differences in adaptations, Young further showed that the benefit of local altruism can be stored in the form of offspring quality and thus promote the evolution of altruism even if the population does not fluctuate. This is because local competition among more individuals resulting from local altruism increases the average local fitness of the individuals that survive. 24. Another explanation for the recognition of genes for altruism is that a single trait, group reciprocal kindness, is capable of explaining the vast majority of altruism that is generally accepted as good by modern societies. The phenotype of altruism relies on recognition of the altruistic behavior by itself. The trait of kindness will be recognized by sufficiently intelligent and undeceived organisms in other individuals with the same trait. Moreover, the existence of such a trait predicts a tendency for kindness to unrelated organisms that are apparently kind, even if the organisms are of another species. The gene need not be exactly the same, so long as the effect or phenotype is similar. Multiple versions of the gene, or even mean, would have virtually the same effect. This explanation was given by Richard Dawkins as an analogy of a man with a green beard. Green-bearded men are imagined as tending to cooperate with each other simply by seeing a green beard, where the green beard trait is incidentally linked to the reciprocal kindness trait. 10. Multi-level selection theory. Kin selection or inclusive fitness is accepted as an explanation for cooperative behavior in many species, but the scientist David Sloan Wilson argues that human behavior is difficult to explain with only this approach. In particular, he claims it does not seem to explain the rapid rise of human civilization. Wilson has argued that other factors must also be considered in evolution. 25. Wilson and others have continued to develop group selection models. 26-27. Early group selection models were flawed because they assumed that genes acted independently, but genetically based interactions among individuals are ubiquitous in group formation because genes must cooperate for the benefit of association in groups to enhance the fitness of group members. 16. Additionally, group selection on the level of a species is flawed because it is difficult to see how selective pressures would be applied. Selection in social species of groups against other groups, rather than the species entire, seems to be the level at which selective pressures are plausible. On the other hand, kin selection is accepted as an explanation of altruistic behavior. 28. Some biologists argue that kin selection and multi-level selection are both needed to obtain a complete understanding of the evolution of a social behavior system. 27. In 1994, David Sloan Wilson and Elliot Sober argued that the case against group selection had been overstated. They considered whether groups can have functional organization in the same way as individuals, and consequently whether groups can be vehicles for selection. They do not posit evolution on the level of a species, but selective pressures that winnow out small groups within a species, e.g. groups of social insects or primates. Groups that cooperate better might survive and reproduce more than those that did not. Resurrected in this way, Wilson and Sober's new group selection is called multi-level selection theory. 29. In 2010, Martin Novak, C.E. Tarnita and E.O. Wilson argued for multi-level selection, including group selection, to correct what they saw as deficits in the explanatory power of inclusive fitness. 3. A response from 137 other evolutionary biologists argued that their arguments are based upon a misunderstanding of evolutionary theory and a misrepresentation of the empirical literature. 2. David Sloan Wilson and Elliot Sober's 1994 multi-level selection model, illustrated by a nested set of Russian Matryoshka dolls. Wilson himself compared his model to such a set. Wilson compared the layers of competition and evolution to nested sets of Russian Matryoshka dolls. 30. The lowest level is the genes, next come the cells, then the organism level and finally the groups. The different levels function cohesively to maximize fitness, or reproductive success. The theory asserts that selection for the group level, involving competition between groups, must outweigh the individual level, involving individuals competing within a group, for a group benefiting trait to spread. 31. Multi-level selection theory focuses on the phenotype because it looks at the levels that selection directly acts upon. 30. For humans, social norms can be argued to reduce individual level variation and competition, thus shifting selection to the group level. The assumption is that variation between different groups is larger than variation within groups. Competition and selection can operate at all levels regardless of scale. Wilson wrote, at all scales, there must be mechanisms that coordinate the right kinds of action and prevent disruptive forms of self-serving behavior at lower levels of social organization. 25. E.O. Wilson summarized, in a group, selfish individuals beat altruistic individuals. But, groups of altruistic individuals beat groups of selfish individuals. 32. Wilson ties the multi-level selection theory regarding humans to another theory, gene-culture coevolution, by acknowledging that culture seems to characterize a group-level mechanism for human groups to adapt to environmental changes. 31. MLS theory can be used to evaluate the balance between group selection and individual selection in specific cases. 31. An experiment by William Muir compared egg productivity in hens, showing that a hyper-aggressive strain had been produced through individual selection, leading to many fatal attacks after only 6 generations. By implication, it could be argued that group selection must have been acting to prevent this in real life. 33. Group selection has most often been postulated in humans and, notably, use social hymenoptera that make cooperation a driving force of their adaptations over time and have a unique system of inheritance involving haplodiploidy that allows the colony to function as an individual while only the queen reproduces. 34. Wilson and Sober's work revived interest in multi-level selection. In a 2005 article, 35. E.O. Wilson argued that kin selection could no longer be thought of as underlying the evolution of extreme sociality, for 2 reasons. First, he suggested, the argument that haplodiploid inheritance, as in the hymenoptera, creates a strong selection pressure towards non-reproductive castes is mathematically flawed. 36. Second, eusociality no longer seems to be confined to the hymenopterans. Increasing numbers of highly social taxa have been found in the years since Wilson's foundational text Sociobiology. A new synthesis was published in 1975. 37. These including a variety of insect species, as well as 2 rodent species, the naked mole rat and the Damaraland mole rat. Wilson suggests the equation for Hamilton's rule. 38. RB greater than C, where B represents the benefit to the recipient of altruism, C the cost to the altruist, and R their degree of relatedness should be replaced by the more general equation. RBK plus BE greater than C, in which BK is the benefit to kin B in the original equation, and B is the benefit accruing to the group as a whole. He then argues that, in the present state of the evidence in relation to social insects, it appears that B greater than RBK, so that altruism needs to be explained in terms of selection at the colony level rather than at the kin level. However, kin selection and group selection are not distinct processes, and the effects of multi-level selection are already accounted for in Hamilton's rule, RB greater than C. 39 provided that an expanded definition of R, not requiring Hamilton's original assumption of direct genealogical relatedness, is used, as proposed by E.O. Wilson himself. 40. Spatial populations of predators and prey show restraint of reproduction at equilibrium, both individually and through social communication, as originally proposed by Wynne Edwards. While these spatial populations do not have well-defined groups for group selection, the local spatial interactions of organisms in transient groups are sufficient to lead to a kind of multi-level selection. There is however as yet no evidence that these processes operate in the situations where Wynne Edwards posited them. 41, 42. Raj et al.'s analysis of host parasite evolution is broadly hostile to group selection. Specifically, the parasites do not individually moderate their transmission, rather, more transmissible variants, which have a short-term but unsustainable advantage, arise, increase, and go extinct. 41. Applications. Differing evolutionarily stable strategies. The problem with group selection is that for a whole group to get a single trait, it must spread through the whole group first by regular evolution. But, as J. L. Mackey suggested, when there are many different groups, each with a different evolutionarily stable strategy, there is selection between the different strategies, since some are worse than others. 43. For example, a group where altruism was universal would indeed outcompete a group where every creature acted in its own interest, so group selection might seem feasible, but a mixed group of altruists and non-altruists would be vulnerable to cheating by non-altruists within the group, so group selection would collapse. 44. Implications in population biology. Social behaviors such as altruism and group relationships can impact many aspects of population dynamics, such as interspecific competition and interspecific interactions. In 1871, Darwin argued that group selection occurs when the benefits of cooperation or altruism between subpopulations are greater than the individual benefits of egotism within a subpopulation. 4. This supports the idea of multilevel selection, but kinship also plays an integral role because many subpopulations are composed of closely related individuals. An example of this can be found in lions, which are simultaneously cooperative and territorial. 45. Within a pride, males protect the pride from outside males, and females, who are commonly sisters, communally raise cubs and hunt. However, this cooperation seems to be density dependent. When resources are limited, group selection favors prides that work together to hunt. When prey is abundant, cooperation is no longer beneficial enough to outweigh the disadvantages of altruism, and hunting is no longer cooperative. 45. Interactions between different species can also be affected by multilevel selection. Predator-prey relationships can also be affected. Individuals of certain monkey species help to warn the group of the approach of a predator. 46. The evolution of this trait benefits the group by providing protection, but could be disadvantageous to the individual if the howling draws the predator's attention to them. By affecting these interspecific interactions, multilevel and kinship selection can change the population dynamics of an ecosystem. 46. Multilevel selection attempts to explain the evolution of altruistic behavior in terms of quantitative genetics. Increased frequency or fixation of altruistic alleles can be accomplished through kin selection, in which individuals engage in altruistic behavior to promote the fitness of genetically similar individuals such as siblings. However, this can lead to inbreeding depression, 47, which typically lowers the overall fitness of a population. However, if altruism were to be selected for through an emphasis on benefit to the group as opposed to relatedness and benefit to kin, both the altruistic trait and genetic diversity could be preserved. However, relatedness should still remain a key consideration in studies of multilevel selection. Experimentally imposed multilevel selection on Japanese quail was more effective by an order of magnitude on closely related kin groups than on randomized groups of individuals. 48. Gene culture coevolution in humans. Humanity has developed extremely rapidly, arguably through gene culture coevolution, leading to complex cultural artifacts like the Gapuram of the Sri Mariamman Temple, Singapore. Gene culture coevolution, also called dual inheritance theory, is a modern hypothesis applicable mostly to humans that combines evolutionary biology and modern sociobiology to indicate group selection. 49. It is believed that this approach of combining genetic influence with cultural influence over several generations is not present in the other hypotheses such as reciprocal altruism and kin selection, making gene culture evolution one of the strongest realistic hypotheses for group selection. Fair provides evidence of group selection taking place in humans presently with experimentation through logic games such as Prisoner's Dilemma, the type of thinking that humans have developed many generations ago. 50. Gene culture coevolution allows humans to develop highly distinct adaptations to the local pressures and environments more quickly than with genetic evolution alone. Robert Boyd and Peter J. Richardson, two strong proponents of cultural evolution, postulate that the act of social learning, or learning in a group as done in group selection, allows human populations to accrue information over many generations. 51. This leads to cultural evolution of behaviors and technology alongside genetic evolution. Boyd and Richardson believe that the ability to collaborate evolved during the Middle Pleistocene, a million years ago, in response to a rapidly changing climate. 51. In 2003, the behavioral scientist Herbert Gintis examined cultural evolution statistically, offering evidence that societies that promote pro-social norms have higher survival rates than societies that do not. 52. Gintis wrote that genetic and cultural evolution can work together. Genes transfer information in DNA, and cultures transfer information encoded in brains, artifacts, or documents. Language, tools, lethal weapons, fire, cooking, etc., have a long-term effect on genetics. For example, cooking led to a reduction of size of the human gut, since less digestion is needed for cooked food. Language led to a change in the human larynx and an increase in brain size. Projectile weapons led to changes in human hands and shoulders, such that humans are much better at throwing objects than the closest human relative, the chimpanzee. 53. In 2015, William Jaworski and colleagues surveyed the opinions of anthropologists on group selection, finding that these varied with the gender and politics of the social scientists concerned. 54. In 2019, Howard Racklin and colleagues proposed group selection of behavioral patterns, such as learned altruism, during ontogeny parallel to group selection during phylogeny. 55, 56, 57, 58. Criticism. The vast majority of behavioral biologists have not been convinced by renewed attempts to revisit group selection as a plausible mechanism of evolution. 59. The use of the Price equation to support group selection was challenged by Van Vielen in 2012, arguing that it is based on invalid mathematical assumptions. 60. Advocates of the gene-centered view of evolution such as Dawkins and Daniel Dennett remain unconvinced about group selection. 61, 62, 63, 64. Dawkins suggests that group selection fails to make an appropriate distinction between replicators and vehicles. 65. The evolutionary biologist Jerry Coyne summarizes the arguments in the New York Review of Books in non-technical terms as follows. 64. Group selection isn't widely accepted by evolutionists for several reasons. First, it's not an efficient way to select for traits, like altruistic behavior, that are supposed to be detrimental to the individual but good for the group. Groups divide to form other groups much less often than organisms reproduce to form other organisms, so group selection for altruism would be unlikely to override the tendency of each group to quickly lose its altruists through natural selection favoring cheaters. Further, little evidence exists that selection on groups has promoted the evolution of any trait. Finally, other, more plausible evolutionary forces, like direct selection on individuals for reciprocal support, could have made humans prosocial. These reasons explain why only a few biologists, like David Sloan, Wilson and E.O. Wilson, no relation, advocate group selection as the evolutionary source of cooperation. 64. The psychologist Steven Pinker states that group selection has no useful role to play in psychology or social science, since in these domains it is not a precise implementation of the theory of natural selection, as it is, say, in genetic algorithms or artificial life simulations. Instead, in psychology, it is a loose metaphor, more like the struggle among kinds of tires or telephones.